name's Peter Bartlett. I'm the Associate Director of the Institute. Thanks for joining us for this Richard M. Karp Distinguished Lecture. We established this series to celebrate the role of the founding director of the Institute, Dick Karp, in theoretical computer science. The series features visionary leaders in TCS and neighboring areas, and it's geared towards a broad scientific audience. And we're grateful to the many contributors to the Richard M. Karp Fund who have made the series possible. I'm delighted to welcome our speaker today, Caroline Uhler. Caroline's the Henry L. and Grace Doherty Associate Professor at MIT in EECS and IDSS. And she's the co-director of the Eric and Wendy Schmidt Center at the Broad Institute of MIT and Harvard. As well as degrees from the University of Zurich and, from the University of Zurich, Caroline has a PhD in statistics from UC Berkeley. She came here on a Fulbright Award. And she was also a research fellow in the very first semester that we had at the Simons Institute in the program on theoretical foundations of big data analysis. Caroline's made important contributions in machine learning, statistics, and computational biology, in particular in causal inference, in generative modeling, and in applications of these to genomics. She's been awarded the Sloan Fellowship and the NSF Career Award, and she's a Simons investigator. And this semester, she's one of the organizers of the semester program on causality, and earlier she was a visitor for Berkeley for that program. So we're very pleased to have Caroline speaking today on causality and autoencoders in light of drug repurposing for COVID-19. Welcome, Caroline.
Thank you so much, Peter. And I'm really excited to be speaking here in this Richard Karp lecture. I still very fondly remember, in my first year during my PhD, when I took his course, in fact, on algorithms in molecular biology, and he really showed me how you can really very nicely marry your love for foundational developments in computer science with really going after important questions in biology. And that's what I'm trying to do also for my own research and my group's research. And so that kind of also already gets me started in terms of where I want to go today. So I will start off by, and I started thinking for this course about, for this lecture, about, if I were to just choose one particular problem in machine learning that, really needs theoretical developments, in what would it be in order to have the most impact on biomedical kinds of questions. And what I want to propose that to be is something that we'll hear term causal representation learning. And I want to discuss why causal representation learning, what could causal representation learning even mean, and what are different kinds of approaches to get at causal representation learning. But really, I'm mainly going to ask questions in this talk and not provide too many answers, and trying to connect that to research that is going on in causality right now, and trying to show different kinds of opportunities for, where theoretical developments in causality could really help to answer and to lead to new approaches in the biomedical sciences. Okay, so first, why is there a need for something that I'll here coin causal representation learning, and then talk a little bit more about what I mean by it. So first of all, why representation learning? So that's easier to answer. So the biomedical sciences, as we all know, has seen this huge, explosion in different data sets. And these are at different scales, right? We have all kinds of genetic information at the DNA level. Then we can measure expression at a single cell level of all of our genes, so all of our 20,000 genes. Then we can get to imaging of tissues, or even imaging and of course medical imaging of whole body parts. And all of these different data modalities have to be integrated in order to get a full picture. Now, often in biology, we don't know what we're going after, right? So we cannot just label these data sets because we don't know what we're going after. So we have huge amounts of unlabeled data. And we know, of these amazing success stories that representation learning has had in different areas of, like, say, for example, integrating captions and images, say, so for text and images. So we're in a similar position here, that we have all these very different data modalities. And of course, we want to find some integrated representations in order to learn the most about what we're actually seeing about the underlying biomedical problems. Now, why causality, and why are just the standard representation learning tools that have been developed in machine learning for other types of questions, why might they not be sufficient? So I think in the biomedical sciences, you actually often not just care about prediction, but really about more than just prediction. So let's think about, your standard machine learning applications that motivate a whole lot of development, that have motivated a whole lot of developments in machine learning, say like recommender systems, right? In a recommender system, I don't really care why, if I'm interested in buying sunscreen, I might also be interested in buying ice cream, right? But you just only care about that that these recommender systems have a high prediction accuracy. I really don't care about the understanding the underlying question of why that is maybe because the weather is good right now, and that's why I might be interested in both of these. But if we're thinking about biomedical questions, and here is a very well-known melanoma data set, which you might be familiar with, where you have like a whole collection of benign and malignant skin lesions, and you may want to understand better what makes it benign and what makes it a malignant skin lesion. Now, in this particular data set, the benign class is actually overrepresented for these bandages, or different types of bandages. And it turns out that the malignant class, and this is what we showed here, is actually overrepresented for these measuring bars. So you can imagine, right, if you just train your standard neural network on these types of problems, well, it will be using whatever you give it. And in particular, it will make use of these kinds of features and will therefore do very poorly in downstream tasks if you're applying this in a different kind of, say, hospital setting. And it's doing poorly because it's not using causal features, right? It's not, these features here are not causal and therefore will not be transportable to a different kind of environment. So you really care about, not just for understanding the underlying biology and really understanding the underlying, medical question of like what makes this different. That's of course one motivation, but also just for being able to really apply these kinds of tools in real biomedical settings. So that leads me to, thinking about that we need something that combines the two, right? Where we kind of can find representations that are not just predictive, but actually causal. And I think what is really so exciting, and I'll talk about different ideas, and I'll just be presenting mainly 3 ideas in this talk for causal representation learning. But I think what is so exciting in biology and the biomedical sciences is that there is this huge hammer that we can use in order to get at something like this. So I think there are some fields that are now getting there as well, but unlike most other fields, what you have in the biomedical sciences for getting at causality is this huge hammer that you can actually perform large-scale perturbations. So we can, for example, knock out any gene we like, so meaning just set the expression of these genes to 0, right, using CRISPR technologies, for example, and see what happens if you do that. So you can intervene or perturb, or in the causal language, intervene in the system in such a way, and in basically many different ways, right, of knocking out, say, different kinds of genes and actually seeing what happens to the system. Or you can apply a drug that's also an intervention or perturbation in the system and see what happens downstream. And of course, interventions are really useful in order to get at causality, right? Think at this particular picture here where, say we want to establish whether good weather causes you to eat more ice cream or the other way around. Well, if you can intervene on the system and say you go to a city and you just give out free ice cream, you'll see that the weather doesn't change, and then can establish that, the ice cream is definitely downstream from good weather, right? So these interventions can really help you in order to establish something about causal directions, and should be really helpful if we're thinking about getting at causal representations. So that's the motivation at a high level from a computational perspective. Let me give you also very particular, or a particular view on, that can kind of unify many of the problems that people are really working on nowadays in single cell biology, and try to put them into a unified framework, and then try to see why causal representation learning might actually be what we need in order to make progress on these different types of questions in single cell biology. So here are like 4 types of questions in single cell biology that, many of the problems that people are studying now can be put into. And let me go through these, and you see here that, I have 2 different levels. So this will tell you already something about causality. So we'll have here just the observational level, and here we have actually the level where you do have interventional data or perturbational data. So let me just start up here and tell you just some questions at the observational data. And this is related to what I said at the beginning, that, you know, in the biomedical sciences, we now often have very different data modalities available to us. So here, and I'm talking here just about the single cell data sets, but the same questions arise also if you're thinking about the tissue level or also the organism level. So for example, you know, data sets that are very widely available are single cell imaging data sets. So here you see each one of these pictures is a picture of a single cell. And you can also get single cell sequencing data sets. So here, for example, every row here is one cell, and you get to measure the expression of all of our 20,000 genes. Now, what you would really like to be able to do, and this is at the cell level and also at the tissue level, is you would really love to be able to translate between them, right? I give you an image like this. Could you predict what the expression profile is? And the other way around, but maybe more interested, you're really of going from images to expression, just because images are much, much cheaper to obtain and also less work intensive. So something like this is something that you would really love to be able to do. And so there is a lot of, you know, questions about integrating different data modalities, but also translating between different data modalities. So this is a question at the observational setting. I get observations of single cells where I take some for imaging and some for sequencing, and let me try to be able to integrate or translate between them. And very similar are questions about moving between different time points. I don't want to spend too much time on here, but you know, these are also questions about, you know, I have some snapshots of data sets, say during cancer progression at different time points, and these different you know, these processes are often so long that you cannot just watch these cells as they go throughout time. So you would like to be able to move forward and backward in time, which is very similar to the question here of different data modalities. So that's at the observational level. And you see like all these questions, just putting them in a very unifying framework of just as transport problems in single cell biology. What about when we also have interventional data? Okay, so when you have interventional data, then here I have two different types of questions that, you know, basically, I think encompass like most of the questions that people would be excited or interested in trying to solve. This one here is the first one that I have here, is also known in the causal literature as causal transportability. So what is this problem? And it's basically at the forefront of everything that people want to do in the pharmacological industry, is say you have like different interventions that you have tested on, say in mouse, okay? So here are all my drugs, and I have performed these drug screens in mouse. Well, what you would really love to be able to do is this causal transportability problem of, I know what the effect is of these drugs in mice. Well, can I predict the effect of these drugs in humans? Right? So let's think of this in the policy setting, right? That's where this problem has mainly been studied. I have like a particular policy that I have introduced in a particular city. Well, can I then predict the effect of this particular policy in a different city by understanding the differences between these different cities? So this problem is known as causal transportability. I try to predict the effect of an intervention that I have seen in one context, try to transport that to a different context and predict its effect. So this is, of course, an extreme case of trying to go from mice to humans. But the problem, and I'll get back to this, you know, a bit less extreme, is I have performed this large drug screen, or also genetic screen, whatever you want to do, different types of interventions on different kinds of cancer cell lines. Okay, so these are actually available data sets. And now here comes a new disease like SARS-CoV-2 or COVID-19. Now you have a new cell line, namely SARS-CoV-2 infected lung epithelial cells. And, you know, you would prefer not to have to redo this whole huge screen, right, on all of these different thousands and 10,000s and 100,000s of different drugs and interventions that you have tested in the cancer setting. But you would love to be able to predict, you know, what will these drugs actually do on SARS-CoV-2 infected lung epithelial cells. If you can predict that, well, then you may be able to rank your drugs, right, based on which drug, we call this drug signature. What is the drug signature, and which drug signature may be most anti-correlated with the disease signature, because this might be a good drug that may be able to move you back from the disease state back to the normal state. Okay, so these questions, you know, you can also move them to the less extreme setting of like trying to go from one disease context to actually a different disease context. Here is a different causal transport question, which is to move between different interventions. So here again, I have, here many different types of interventions. Say I have knockouts, and, humans have 20,000 genes. Nowadays, you can do screens where you knock out one gene after the other. So for all these 20,000 genes, you can actually do that. But, you'll never be able to do all combinations of knockouts, right? I mean, even if I look at 20,000, choose 5, and then I want to do 20,000, choose 3, etc. I'll never be able to do any, all of these experiments. So at some point, you need to be able to predict the effect of new interventions or new combinations of interventions. Or if you're thinking about the drug context, right, I have done a drug screen on some molecules. Well, I have new molecules, and I would like to be able to predict what will happen if I actually, if I apply this particular new molecule to my cells. So this is it's another causal transport question, which is transporting between different types of interventions. Okay, so here I'm transporting between different contexts. Say I have some perturbations, I've seen them in one cell type or one disease context. I'd like to be able to predict what happens in a different disease context. And here I have seen some perturbations, some interventions. I'd like to predict the effect of new interventions. So this is, encompasses a lot of, as you see here, right, a lot of important questions, say in the pharmacological setting, or here, in the basic biological setting of really trying to really understand how different data modalities are related to each other. And what I want to do now is, of course, here it kind of already seems clear that something like causal representation learning will be critical, right? We care about causality because we care about predicting the effect of an intervention, which only has an effect downstream of it. And some type of representation learning will be critical because we have all these different kinds of data modalities that we need to integrate. But now let me first maybe take a step back and ask, how would these questions, more traditionally be looked at, okay? And, traditionally, if you try to predict the effect of a knockout, say, over a particular intervention, then how biologists have always pictured this and what the ultimate goal was in biology is to really get at completely the underlying mechanistic model of how, what is actually happening in a cell. So how are genes interacting with each other, which gene turns up or down which other gene, and really learn these, very large scale genetic networks here, where the nodes are genes, right, and the edges tell you which gene and how it regulates which other gene. And thinking about these networks, so also thinking about how that relates to causality. So Sewell Wright was, was a geneticist, but really developed the foundations of much of the field of causality today, where he comes from studying heredity in genetics, and drew out these kinds of networks, which are causal networks, and tell you, about heredity information in these species. And this is in some sense how we're looking at these gene regulatory networks themselves as well. And let me just give you, what is the kind of framework that Sewell Wright developed at the time. These are these causal structural equation models. It's a very, very intuitive model of how, these networks here can be read, where, if we're thinking about it in the gene regulatory space, my nodes here, these are random variables, these are genes. They're random, there's some randomness because this is the expression, this measures the expression of a particular gene. And it's a very intuitive model where, a node here, so you have this directed graph that corresponds to causal relationships, and a node here is some function of its parents and noise. Okay, so here, for example, x4 is a function of x2 and x3 and some noise. These functions, of course, can be highly nonlinear, which is the case also often in genomics, right? And the noise, of course, doesn't have to be Gaussian, doesn't have to be additive, etc. So, if we're thinking about our problems here on the previous in the previous setting, where, if you try to predict the effect of a knockout, well, if you have the full understanding of this underlying, regulatory network, well, then you can actually predict the effect of a knockout, right? And so, like what we then would want to do, and what I was excited about, many years back when I saw how large these observational single cell gene expression data sets became, is to ask, well, maybe we can use, this kind of framework and try to, from observational data on the nodes, try to actually infer these networks. Right, and then we could solve some of the questions that I asked on the previous slide in terms of predicting what happens if I'm setting one of these genes to 0. So let's talk a little bit about structure discovery and causal structure discovery. And, this is a rich history and is still very active field today. And I'll give, of course, some more references afterwards. But, here it's really started by very foundational work by Peter Spertus of telling us, of course, we know, as I already said at the beginning, right, if you just have correlations like good weather and ice cream, then just from observational data itself, we cannot tell which direction it's going, but you can learn some of the underlying directions, even just from observational data. And, Peter Spertus, proved results about what can be identified and also provided algorithms for actually learning these causal graphs just from data itself on the nodes. So, if we try now to think of like, well, then, that's exciting, right? Can we just apply this and learn these gene regulatory networks from data on the nodes? That led us down to really asking about, how good do these algorithms perform and what are their assumptions needed in order to provide the correct graph when you have just observational data on the nodes. And what is really interesting is, a fundamental assumption behind these algorithms for causal structure discovery is something known as faithfulness, which says the following, that, here, if we're just looking at 3 genes, and, gene 1 could have a positive effect on gene 2, so upregulate gene 2, but gene 1, could also have a negative effect on, say, this gene 3, and then gene 3 upregulates gene 2. So, of course, it could happen, and these things do happen in biology, that, different kinds of paths, the effect along different paths can cancel each other out. Now, the faithfulness assumption, what it assumes is that this doesn't happen, okay? That paths along different paths do not cancel each other out, because it's a problem when you have that happen and you're trying to learn the structure, because now, because they cancel each other out in the data, it looks like gene 1 and gene 2 are independent of each other. And so if they're independent, then you would remove some edges between them, like this one here, right? And that would lead to a wrong graph. So that's a faithfulness assumption that says that these paths that they are not allowed to cancel each other out. Now, of course, if you have infinite amount of data, then the probability of this happening, right, that this exactly cancels each other out, well, that has measure 0. So you can say like, well, maybe I don't care about these types of assumptions. But think about when you actually have, sample sizes that are not infinite, right? How do we actually do independence testing? Well, we have some threshold where we're saying like, well, I'm going to decide that these 2 genes are independent if their correlation is small enough. But as soon as you're you're adding a threshold, then of course this assumption doesn't have measure 0 anymore. And so that's what we had quantified, in earlier work, and shown that, this assumption is actually a very strong assumption when you have finite sample sizes. And, it will be so strong that even like if you have like 10 node graphs and, you have quite large sample sizes, like even 1 million samples, then even for very sparse graphs, this assumption basically doesn't, with probability 1 doesn't hold, meaning that in these kinds of cases, you will, for the algorithms that assume faithfulness, and, and I will get to like questions about, is this like, do you really need this assumption or not? But something like this is what is needed. We still don't know whether in fact the really minimal assumptions needed, but something like faithfulness is needed. So this just means that with the, then these algorithms will not get the graph right. So you need huge amounts of data in causality for just learning the underlying causal graph. So for performing structural learning in causality from just observational data. So that's a problem, of course, and that was when I moved to MIT, in fact, and when I got introduced at the Broad to the kinds of data sets that would be coming out soon, and what got me really, really excited. So I was very discouraged when I was working on, single-cell RNA-seq data sets and trying to learn these gene regulatory networks. Now, what was being developed at the time is like these large-scale perturbational screens, right? That you can actually now go in and intervene on any gene you like. And this should give you more information, as we said before, right? If you go out and provide everyone ice cream and you see that nothing changes upstream, that the weather doesn't change, then you know that, the weather is certainly not downstream of your perturbation, right? So perturbational data should give you a whole lot of more information. And so this then motivates the question of asking, from a theoretical perspective, well, how much more can you learn when you also have interventional data together with observational data? Might that maybe get you closer to the kinds of to algorithms that you could actually use in this genomic setting in order to learn these gene regulatory networks? And there is a lot of literature here in this for causal structure discovery from interventional data. Here I'm just stating some of it that we were very excited about it early on, building on Frederick Eberhard's formalism that he had developed of like how to even think about interventional data sets, and then developing algorithms like these over here that can really combine observational and interventional data in order to learn these causal graphs. Now, I always want to highlight people that are actually at assignments, long-term participants here. There's a lot of people working in this area, like Sarah Malyakane and Daniel Malinsky, currently there. These algorithms, they do work, of course, better than just when you have only observational data. But here I'm just showing some result to show you that we're still far away from doing anything that would really get us to gene regulatory networks. I mean, here I'm showing just graphs on 24 nodes with huge numbers of samples. And, these false positive, true positive kinds of curves here are not something that we're happy with in order to really learn these gene regulatory networks. And I'm very excited about more theoretical computer science work that people have started nowadays with Arnab and Costas, who are at the Simons Institute, who are really thinking more deeply about, how much more information does actually can interventional data provide you as compared to observational data, really getting a theoretical framework for thinking about these kinds of trade-offs, and also sample complexity results of how much data you really need in order to just estimate the parameters in these causal graphs or really learn these graphs as well. Okay, so that's kind of where we are, I think, in terms of, trying to learn these gene regulatory networks from a mix of observational and interventional data. And so that brings me back to this kind of question, right? So here, yes, one approach is, and this is the standard approach, is to try to really learn this underlying gene regulatory network and then try to answer these types of questions. What will happen if I knock out a particular gene? But, there is a question of whether this is really necessary, right? Is it really, I mean, if these are the questions that I in the end want to answer, do I really have to go this full route of actually learning the underlying gene regulatory network? And so that's what I want to be talking about now, and maybe give you some ideas of like, maybe, we can get around it without actually going this whole path. Okay. Now, I'm talking about causal representation learning, and what you need, or what we want to do here is, of course, we need to somehow be able to generate data corresponding to a new intervention, right? So I have some interventions that I've seen here, and all of these questions, they're all generative kinds of questions. I want to be able to predict what happens when I'm performing a particular intervention in a new context, or here, what happens for a new type of intervention that I have not yet seen. So as you see everywhere here, I'm trying to generate data corresponding to that intervention, corresponding to a new modality, corresponding to a new time point. So definitely what I'll need is some type of generative model. And what I will be using here are autoencoders, just because we understand them quite well, I think, or the types of things that we need to understand we have, or starting to understand at least. So, we all know about the amazing success stories of neural networks and how they've, being used in the standard discriminative modeling setting. What I'm going to talk about here and what we need, right, for these types of questions are to really apply them in the generative model setting. So where we want to generate new data set, a new data point corresponding, say, to a new intervention. Now, autoencoders are, a form of nonlinear principal component analysis where you're using a neural network in order to embed from your data set, say images or sequencing data, to some lower dimensional latent space. And then you have another neural network that maps you back to the original data modality. And these are trained by just minimizing the reconstruction error here. Okay, so these are, quite simple in the sense, but so always used for dimension reduction. Now, what you'll see in this talk, and it might sound a little bit crazy at first, is that I'm going to use autoencoders in a non-standard way. In fact, I've never used in this way. namely where I'm not using autoencoders for dimension reduction, but I'm going to use autoencoders for embedding the data into a higher dimensional latent space than what you started off with. Okay, so this may sound crazy and might sound like something, why would you ever want to do something like this? Because now you have enough parameters in order to just learn the identity map here. And yes, that's the case. Okay, and so maybe that means that I'm not learning any interesting thing in this latent space. But I think we have work here where we're actually characterizing what are the functions learned when you're going through these, larger dimensional latent spaces. And, there is a lot of motivation for why to look at larger, wider and wider neural networks, because they have seen, at least in the standard classification setting, to perform actually very, very well. Okay, so that motivates for us, or had motivated us at that point, to actually look at autoencoders where we're going to a larger dimensional latent space. And I'll tell you a bit about afterwards, like how this might relate to causality. Okay, but just already here to provide, so what I mean by an overparameterized autoencoder is where I'm going to use a larger dimensional latent space in order to embed my data. So I'm not using autoencoders for dimension reduction. Okay. So, and now what I want to do is just give you 3 ideas of, what might we be able to talk about these types of problems without going through the whole route of actually going to, first learning the whole underlying causal graph, and then through that trying to predict the effect of one of these interventions. And also maybe let me make the problem also a little bit more difficult and, say what I also want to be able to do is like use other kinds of modalities where it's not even clear what the nodes of my graph are, right? So before my network, it was like genes, and I tried to learn this gene regulatory network. So if you have expression data, well, that's data on the nodes. But what about if I give you an image like this from a cell, right? So it's certainly not clear what the causal variables are. In fact, the pixels are definitely not causal variables. So how can I even think about causality when I have things like single cell images? Okay, so that brings me kind of to the first idea, and let me just discuss this. And it also, in fact, so where it comes from is actually to solve this kind of problem of like, how do I use even data from different kinds of data modalities, and how can I go between different types of data modalities, and how might that help me actually get at causality? Okay, so what am I proposing to do here? So we have these very different types of data modalities, and let me just concentrate on 2 of them, which is, what we have seen before, if single cell images and single cell RNA-seq, so expression data on the other side. Well, we would like to be able to translate between them and integrate them, right, into one common representation that captures, as much information as possible. So a very natural thing to do is to say like, well, let me do an autoencoder that goes, from images to a latent space back to images, and an autoencoder that goes from expression data to the latent space back to the expression data. And let me then just, because, in my data set, I just at random took out some cells and sequenced them, and I took at random out some cells and imaged them. Well, it's the same population of cells, right? So let me then enforce that in the latent space, it's the same distribution. I can do that by, adding a discriminator, say, in the latent space, so that I cannot distinguish cells that were imaged from cells that were sequenced, okay, just because it's the same distribution of cells. And in this way, I can translate between the different data modalities. But even more, what I want to propose here is that different data modalities might actually also allow you or help you get at what the causal variables actually are. And so why is this the case? And here I will, I'll defer and also relate to, of course, other work in causality, where the invariance principle has been used. But let me first explain what I'm thinking of here. Well, so if you want to get at what are the causal features, well, a feature is more likely to be causal, right? It is if it is invariant to how you're looking at it. So in this joint latent space here, right, where I have like my features that are independent, that are invariant to whether I'm looking at it from an imaging land, or whether I'm looking at it from sequencing land, or from any other land, right, then it should be something that is more intrinsic to the cell itself, and hopefully more causal. And hopefully the more data modalities you actually have, the more different views you have on the system, the more you should be able to actually get at these causal features. So that's the intuition or the motivation here. There is, of course, a whole lot of theory to be done here, but that's at least the motivation or the intuition for why these latent spaces should help you to get a causal features. Now, there is a lot of work on using invariances with respect to interventions for getting at causal relationships. What I'm proposing here to do is to get at causal features. And let me also relate to other work that is trying to do a causal feature learning, which is a different type of approach without using different types of modalities, but really using either disentanglement in the latent space or using interventions to figure out features that are moving together and therefore should be causal features. So I think it is a really exciting space here of somehow combining different approaches to thinking about what causal representation learning could be in terms of causal feature learning, and how maybe different data modalities can help, how having access to interventional data could help, and more generally, this principle of invariance could help to either get a causal relationships, so these edges between causal nodes, or could even help you to get at these causal features themselves. Okay, so that's a high-level idea of how these different data modalities might actually help you. So if I go back here and also ask myself, so often this is what is called causal representation learning, that the end goal would be to actually identify the causal features. So I would say that I will propose that causal representation learning should actually be able to do more than just this. It shouldn't just be a representation where your features are causal or causal variables, say, but it should also allow you to solve problems like these causal problems that I have down here, of predicting the effect of an unseen perturbation, unseen intervention in the latent space. So the latent space should have that information that I can, apply interventions and try to predict what they will actually do. And, how can we get there? And how can we even think about this question of predicting the effect of unseen interventions? Let me get to the second idea. And this is based on Julia Pearl's causal hierarchy, okay, where I really like this picture where you have at the bottom, your standard association and standard things that neural networks are good at, finding correlations, for example, how is my headache, how is like taking an aspirin and having a headache related to or associated with each other? You have the second level here of interventions, which asks, what will happen if I take an aspirin? And all the way at the top is counterfactuals, which is in fact what all of causality theory today is concentrated on, and allows you to answer really difficult questions of, what would have happened had I not taken the aspirin? Okay, that's this counterfactual level, all the top here, which is what causality is all about. And so in order to answer these types of questions, you really need to know this full underlying causal graph. Okay, so causal structure discovery and then, estimating all these causal parameters allows you to answer these types of questions here all the way at the top. Now, what I want to propose is, and Julia Pearl would say this is at the Neanderthal level here, but I'm actually very happy with this Neanderthal level. And I would like to propose that, in biology, we actually often care about this second level and not so much about the counterfactual level. So unless you're studying, say, clinical trials, but otherwise, all the questions that I presented about predicting the effect of an intervention, they're all here, right? There are questions like, well, what will happen if I knock out genes 1, 2, and 3? Or what will happen if I apply this new molecule that I have not yet seen in my data set? All of the questions I talked about are at the second level. And in biology, you don't actually, in the basic biological questions, you don't care so much about, or in science more generally, about counterfactuals, right? Because I want to do these predictions and then be able to test these predictions. Anyways, I'm going to go to the lab and afterwards actually validate whatever your predictions are. However, these questions are not your standard prediction problems like what you have here in the association level. They are causal prediction questions, right? Because I'm trying to predict the effect of an intervention, but they're not all the way up here. So it is, to me, reasonable to believe that maybe, all of this, like what we're doing right now of like trying to learn all these causal graphs and getting at these gene regulatory networks, well, maybe you actually don't need that in order to answer these questions about predicting the effect of an intervention. So there is, I think, a really huge opportunity, and there is a need for coming up with the right theory of understanding what is really needed in order to answer causal prediction problems that are not counterfactual questions, right? Which is what we have all of the theory around right now. So what is the difference between these two levels, and how much less is needed in order to answer these causal prediction questions here at the second level? And these are really the questions that we need to do in drug discovery, right? Or in all of these causal transportability questions that I had there, where we have here all of the different perturbations or interventions. Here are, say, my cancer cell lines, and I want to be able to fill in these white spaces here of what will this drug do in this particular cancer context. Or I add a new column here, which is now COVID-19, and I want to predict what will all these drugs actually do in this particular setting here, where I have not yet seen any of these interventions. So that's just a call for, that I think what we really want is some theory here, and that it is very conceivable that we actually don't really need to go all the way to learning these causal graphs in order to answer these types of questions. And let me end with like a third idea, and an idea of like, how could we go about this? Okay, so and as I said, I think in causal representation learning, I want to have representations that can actually help me, that can not just identify causal features, but that could answer these types of questions here at the second level, of having a representation where I can actually predict what the effect of an intervention is in a context where I have not yet observed it. Now, if you put on your machine learning hat, I'm going to propose something that is very, probably very natural of like how you would maybe go about answering these types of questions. And, so here is how I'm thinking about it. Well, we have our latent space, and you have, say, some drugs or some other interventions, some intervention that you have already observed in one context. Okay, so say here I have a particular drug, I have a particular cancer type, and I know what this particular drug does in this particular cancer type. So, let me go to the latent space. I have here my cell type, my disease, my cancer cell type. I add the drug, and this is what comes out. Okay. Now, let me get data from COVID-19, and here is my, my SARS-CoV-2 infected lung epithelial cells, and I would like to predict what this drug does on these SARS-CoV-2 infected lung epithelial cells. If I can do that, then I can rank my drugs, right, as I explained at the beginning, and rank them based on, and try to figure out which drug signature is most anti-correlated with the disease signature. Okay, so how can I do something like this? Well, we all know these nice Yosemite pictures of Yosemite in the summer and transport it to Yosemite in the winter, or all these Word2Vec kinds of really amazing, amazing applications of generative models. And here I'm doing it, not using GANs, but actually using autoencoders. But how these things have shown to work quite well is you just take the vector in the latent space, you take this vector corresponding to adding a drug, and you just move it to your new cell type, your new disease context, and predict that this is what is, and here is the prediction, but you're moving it back into your expression land, and this is the prediction of what you think this drug will do. Now, this is such a simple idea, and it has been used in pharma. It has been tried in the pharmacological industry, and it has not worked as well. Okay, so here, and you can just test how well it works because you have these kinds of data sets, so you can actually test how well it works. And what I think we need, and here is just one starting idea of how we can maybe go there, and how we have at least seen success in terms of like just this causal transportability problem of trying to predict the effect of an unseen perturbation. What we have actually seen work very well, in particular in the SARS-CoV-2 setting. But this is just an initial idea, and of course there should be a whole lot more research. It's just trying to get this type of research started, is to identify, what are inductive biases of representation learning algorithms that are well aligned with trying to answer or trying to get at these causal transportability questions. And, I did mention these overparameterized autoencoders for a reason at the beginning, Because, and it's very intuitive to use something like an over-parameterized autoencoder, because what do you need in order for these kinds of causal transportability problems to work out? Well, you need that the effect of a particular drug in the latent space is aligned, right, across different contexts so that you can just move it around. And, out comes the prediction of what this drug will do in the new context. Now, if you have more space in the latent space, right, instead of less space, how these autoencoders are usually used, well, then you have more space and it's much easier in order to align the effect of drugs as compared to if I'm going to a more smaller dimensional latent space where everything will be even more crumbled up, right? Even if your effect was linear before, now it will become more crumbled up just because I'm going to a lower dimensional latent space. But, this is just one approach where we have some theory about why this is actually working out and when is it working out. But I think more generally, I think what we need in order to get to something like causal representation learning is to really understand and develop the types of representation learning algorithms whose biases, whose inductive biases will be aligned with the types of causal questions that we're trying to answer. In particular, I feel that these are causal questions that we should be able to answer in the latent space of what will happen. Can you predict the effect of an unseen intervention that maybe you have seen in one context, or can you predict the effect of new combinations of interventions? Hopefully, it would be something that you can add up the effect of single interventions, or maybe you want to find some other latent spaces. But those are the types of questions that I think causal representation learning should allow you to answer. Okay, so these were 3 high-level ideas. Here is just to show that this has in fact worked for predicting the effect of drugs in the SARS-CoV-2 setting. And what is quite interesting and we're excited about is the drugs that actually came out just entered so one of them, in fact, the top drug just entered phase 2 clinical trials, and we'll see what actually happens with this one. But let me end with this and just tell you, looking forward in terms of what will come. Well, if you're able to predict, and hopefully causal representation learning will allow you to do something like this, to predict the effect of unseen interventions, well, what you can then do is answer amazing questions, and these are things that we're very excited about, of what would be the optimal interventions in order to move you from one state to another. And states can be move you between different cell types, so reprogramming cells. It can be moving you from a disease state to a normal state, et cetera, et cetera. It can be, moving you from an age state to a young state. So a lot of questions in biology that are really fundamental questions are, cell state transitions and figuring out what are the best interventions in order to move you from one state to the other. And I think there is already a lot of research in the space of, experimental design of interventions. And of course, a whole lot more need for new theory in this setting where you can perform interventions, you can actively learn from interventions, and you want to really identify the optimal perturbations or the optimal interventions in order to move from one state to a particular other state. And hopefully something like causal representation learning will help you to do that in this latent space where you're actually trying to figure out how to move around in order to get from one state to the other. And with that, let me end. Hope I gave an intuition for what I think causal representation learning should be able to do, how it connects to different kinds of research that is currently going on in causality, and highlighted some of the exciting developments that have been going on there, what is being done now in this program. And I'm ending with thanking, of course, all of this work has been done by an amazing group of people in my group, and then, of course, funding. And thank you all for listening.
Thanks very much, Caroline. Questions? Well, let me start while folks are thinking about that. Maybe you could tell us a little more of the story behind the drug that for COVID that is going into clinical trials two slides back.
Yeah, perfect. So what is happening and what is going into clinical trials is, so here, these drugs that were in this cancer screen, of course, have a whole lot of drugs, and there's more story, but there is a whole lot of drugs that are drugs against in the cancer field that have a whole lot of targets. So these drugs are not the ones that are going into clinical trials. What you want to do, right, if you identify which drugs are your top candidates, and that's actually another use of causality, is to figure out, if you have drugs that have a whole lot of targets, which one is your putative causal target. And in particular, that's what we did by trying to identify, based on these targets that we found here for these drugs, which one is the one that is most upstream of all of the differentially expressed genes. What comes out from that analysis is that it is RIPK1, which is here. And what is currently in clinical trials is an inhibitor that is specific to RIPK1. So to this putative target that we identified based on these drugs, that is actually the most upstream of all of these different differentially expressed genes. In fact, it also turns out that this particular protein directly binds to SARS-CoV-2 proteins, which is another kind of exciting thing.
Fantastic, thank you. Any other questions? Do we have any questions on Zoom? We actually have a... All right, please join. Oh, yes.
Following up your question. So what do you mean by upstream about this gene? Like, were you? So I thought you avoided doing like sort of like a whole network construction, and just learn an implicit representation, and that's how you achieved this. But now, when you're talking about upstream, did you really go back to causal discovery algorithms?
We indeed went back to causal discovery algorithms. And try to identify which ones, in fact, using exactly the kinds of tools that I told you about at the beginning. We exactly use these types of tools because now we do have a mix of observational and interventional data available. We use these types of tools for identifying, based on all of these genes that we have, the differentially expressed genes in SARS-CoV-2 infection, plus all of these possible targets, to identify which one is, estimated to be upstream of all of the differentially expressed ones. And that's how, independent of whether you're looking at it in different kinds of cell lines, in fact, it's in all of them, it was always consistent that RIPK1 was the one that is most upstream. Yes, we did go back to, I think at the end there needs to be some combination of the two. But you can get hypotheses like this. I think you still need the step often of actually really understanding this the underlying mechanisms, right? The representation doesn't, it would be great if the representation can give that to you. But here we did estimate these graphs as well.
Hi, Caroline. Thanks very much for the great talk. Could you talk a little bit about the representation learning aspect and its relationship to causality? Like, I think there was an implicit use of the fact that good, understanding causal mechanisms is related to good representation. So could you talk a little bit about what types of data you need to have confidence in that the representation you learn will be causally predictive?
Yes. So I think if I can solve, so we have these data sets, right? We do have these data sets where we have different interventions in different contexts. So if you can predict, I think if you have a representation where you can predict the effect of an unseen intervention, then for basically most problems that people are excited about, that's sufficient. So I would define, and I'm just proposing this, I will define that as a good enough causal representation for many of the tasks in biology. And so that's how you would validate it, right? By whatever you want to do, you find your representation, but you can solve these types of questions at this level of being able to predict the effect of an unseen intervention. So I tell you, you tell me what happens when you knock out gene 1 and 3. You can use all of the other data. If you can do that, I think you have found a causal representation.
That is in the validation stage, in the of your model.
You would leave out some of the different perturbations.
So you're talking about validation right now, correct?
Correct. Yep. Did I understand the question correctly? I hope.
Yeah. So I guess I mean, but there is also this thing that you mentioned that this overparameterization somehow relates to whether or not you will pick up a representation that actually has this ability. So
yeah.
Right.
So yeah, I think this is a great, so that's what I'm saying like this is an approach that we have tried. So where, how we're doing this transport is just by moving around vectors in the latent space. That's one way of trying to predict the effect of an unseen intervention. And there we have seen that these overparameterized latent spaces actually help you to do that, to align the effect of interventions that I have not yet seen. But I think it is an open problem. What I would like to see is, any representation learning algorithms where you can define their inductive biases in such a way so that you can solve these types of questions. And I'm just giving one approach that we have been looking into and trying, but I think there is a whole lot to be done there.
So my other question I had is, you mentioned this invariance principle. I was actually wondering how crucial is to use all of the techniques and the reasoning from causal inference to be able to succeed in such out of distribution tasks, instead of just arguing about invariance and just having invariance as our principle and not even going into like elements of causal inference. I'm not saying we should, but I'm just asking, where is it most crucial to use causal reasoning when doing causal representation learning and not just call it invariant representation learning?
Yeah, so to me, where causality comes in is where I'm trying to predict the effect of an intervention. So that is right, that I'm actually trying to answer these types of questions and not, so if you're thinking about your standard representation learning tasks of like, say, captions and images, and I want to like, find something that is, say, invariant, I could also just be asking about questions that are down here of like predicting, say, I don't know, whether there is a cello inside my image, right? Your standard things. But I do want to be able to answer, and this is to me where causality comes in, is when I'm having an intervention, this will only have an effect downstream. So I do need to understand something about the causal relationships. I don't need to go all the way up here, but this is a really different level than down here. And I think invariance can help you, but I think you need to also be thinking about causality. Does that make sense?
All right. Thanks very much, Caroline. Join me in thanking Caroline for a really fascinating talk.
Well, thank you all very much. I'm happy to continue discussions online or via email. Thank you.